Welcome to Bio Bits and Bytes. We've got some really interesting materials on synthetic biology today, and we're going to pull out the key insights you need to get a solid handle on this field.
It's definitely a hot topic.
Absolutely. We've got reports covering the basics, applications in farming, and environmental work, plus some important stuff on safety and how it's being managed in places like the U.S. and China.
Yeah, a real mix.
Our goal here is really to connect those dots for you, give you the main takeaways without, getting lost in the weeds.
Sounds good. Synthetic biology is, well, it's moving fast and understanding it is becoming pretty important.
Totally agree.
Yeah.
So let's dive right in, the absolute basics first. What is synthetic biology? Can you break that down for us, make it easy to grasp?
Okay, let's do it. So synthetic biology. At its core, it's really about taking engineering principles and applying them to biology.
Engineering principles, how so?
Well, think of traditional biology as, studying how life works naturally. Synthetic biology uses that knowledge to actually design and build new biological parts, devices, systems, or even redesign existing natural ones for specific useful jobs.
So it could be just tweaking a microbe or something.
Yeah, it ranges from modifying existing organisms right up to potentially building new biological functions from, well, non-living materials. It's about engineering life, essentially.
Okay, actively engineering, not just observing. Now, the sources mentioned overlap with genetic engineering, biotechnology. What's the real difference here? Is it just ambition?
That's a good question. There's definitely overlap in tools, right? But synthetic biology often aims, well, bigger, more system-level thinking.
How do you mean system level?
So traditional genetic engineering might change one gene, maybe a pathway. Synthetic biology often tries to engineer whole new functions, maybe build complex circuits within a cell, or even, like I said, artificial systems. It's very design-driven.
Oh, right. I saw mention of a design, build, test, learn cycle, like in engineering.
Exactly. The DBTL cycle. Design, build, test, learn. It's that iterative process. The idea is to make biology more predictable, more engineerable, to meet specific human needs reliably.
Okay, that makes sense. Now, you mentioned biological parts. What are those exactly? One guide used a toolbox metaphor. Can you maybe unpack that toolbox for us? What are the key components?
Sure. Think of these parts as standardized, kind of interchangeable units, like building blocks. The sources mention a few key ones.
Right.
Okay, so first, promoters. A promoter is basically the on switch for a gene. It's a DNA sequence that tells the cell's machinery when and how much protein to make from that gene.
Got it. On switch. What else?
Then you have regulatory proteins and RNAs. These are more like dimmer switches or, more complex controllers. They fine-tune gene activity, turning it up, turning it down based on different signals.
And RNAs themselves, they do more than just carry messages.
Oh, yeah. RNAs, ribonucleic acids, are messengers, sure, but they also act as regulators directly. And then there are scaffolds. Think of these as organizers inside the cell.
Organizer.
Yeah, they can bring different molecules together in the right place physically, making the whole engineered system work better. So promoters, regulators, RNAs, scaffolds. That's your basic toolbox for building these complex biological systems.
It really is like a biological toolkit with standardized parts. Now, that DBTL cycle design, build, test, learn one source, mentioned the engineering biology roadmap using this.
Yeah.
Why is that cycle so important?
It's really fundamental to tackling the complexity. Biology is, well, incredibly complex, right? So many interacting parts. The DBTL cycle gives you a structured way to manage that.
How does it work in practice?
You design your system first, say, a pathway to make a biofuel. Then you build it, which usually means synthesizing the DNA you designed. Then you test it, put it in an organism, see if it works. And crucially, you learn from the results. What worked, what didn't. Then you go back, refine the design, and repeat.
Iterate until it works properly.
Exactly. It's about managing that complexity, using design rules to abstract away some details so you can actually engineer these integrated biological systems with many components.
Right, a methodical approach. Let's zoom in on the build phase.
Yeah.
Engineering DNA. How is that actually done? Notes mention single-stranded and double-stranded synthesis.
Okay, yeah. Making custom DNA is key. For single-stranded DNA, it's often done chemically, nucleotide by nucleotide A, T, C, G, building up the strand, usually on automated machines, attaching the growing chain to tiny beads or chips.
Like stringing beads in a very specific order.
Pretty much. Very precise.
And then to get the double-stranded gene you actually need.
Right. For double-stranded gene synthesis, you often make shorter single strands. First, these are called oligonucleotides or oligos.
Yeah.
You design them so they overlap.
Overlap.
Yeah, so they stick together in the right places. Then an enzyme, DNA polymerase, fills in the gaps to make longer double strands. You can amplify this using PCR polymerase chain reaction, which is like a DNA photocopier.
So you can make lots of copies.
Exactly. And for really big genes, there's a technique called polymerase chain assembly that stitches together multiple fragments all at once.
It sounds like building with DNA Lego pieces. So once you have these engineered genes, how do you describe them?
Yeah.
Keep track. The notes mention standardized symbols, SBO.
Ah, yes, SBL, synthetic biology, open language. This is super important for, well, communication and just making the science reproducible.
Why standard symbols?
Imagine trying to share an electronic circuit design without standard symbols for resistors or capacitors. It'd be chaos, right? SBL provides a standard visual language for biological parts.
So symbols for promoters and
Yep. A promoter might be an arrow. A ribosomal binding site, where protein synthesis starts, has its symbol. Coding sequences, the actual gene part, are often blocks.
Hmm.
Terminators, stop signals, have their own symbol. Even things like plasmids, the circular DNA often used to carry genes, have standard ways to draw them.
so everyone's speaking the same visual language.
Exactly. It makes designs clear, shareable, easier to build on. It's like having that common blueprint for bioengineers.
Makes perfect sense. Okay, another huge tool mentioned everywhere is CRISPR. How does that fit into synthetic biology?
Oh, CRISPR is indispensable. It's completely changed the game for gene editing. As the sources say, it comes from bacteria. Originally, it's their immune system against viruses.
And we've repurposed it.
We sure have. Systems like CRISPR-Cas9 let us make incredibly precise changes to the DNA sequence in almost any organism.
How does it get that precision?
Well, you have the Cas9 protein, that's the molecular scissors part, and it's guided to the exact spot you want to cut in the genome by a guide RNA molecule, which you design to match that target DNA sequence.
So the guide RNA finds the address, and Cas9 makes the cut.
Precisely. Then the cell's own repair systems kick in. And we can trick those systems, for example, using homology-directed repair,
Hmm.
HDR. We give the cell a template DNA with the change we want, and it uses that template to repair the cut, incorporating our edit.
Wow, that's incredibly targeted. Okay, so you've designed your DNA, maybe used CRISPR to tweak things.
Mhm.
Where does this engineered DNA actually go? What's host engineering?
Right. Choosing the host organism is critical. It's the chassis where your engineered system will run. Different hosts like bacteria, E. coli is common, yeast, or even cell-free systems have different pros and cons.
Like what?
Well, bacteria grow fast, easy to manipulate. Yeast can handle more complex proteins. Cell-free systems are good for quick tests or making toxic stuff. Sometimes you might even use multicellular organisms or whole communities of microbes. It depends entirely on the application.
So you pick the host based on the job you need to do.
Exactly. Growth rate, protein folding ability, how easy it is to get the DNA in. Lots of factors.
Okay. One last foundational concept, biobricking. What's that about?
Biobricking is a specific method focused on standardization. The idea is to use predefined, standardized DNA parts called biobricks. Each biobrick has a known function like a promoter or a coding sequence, and crucially, they have standardized ends.
Standardized ends.
Yeah, specific DNA sequences recognized by restriction enzymes, those molecular scissors again. By using enzymes that cut at these standard ends, you can easily cut out biobricks and then use another enzyme, DNA ligase molecular glue, to stick them together in new combinations. This creates recombinant DNA.
Oh, so it makes assembly much more modular, like snapping predefined blocks together.
That's the idea. It aims to simplify building complex circuits by using these well-characterized compatible parts, speeds things up.
Okay, that covers a lot of the how. Let's shift gears to the what for applications. Agriculture seems like a big one in our sources.
Huge potential there, definitely, for increasing productivity, sustainability. Several interesting examples came up.
Like the biosensors for crops.
Exactly. Imagine plants that can signal if they're stressed, lack of water, pests maybe by changing color, an early warning system for farmers.
That would be amazing. What else?
Developing novel antimicrobials for livestock using CRISPR. This could target harmful bacteria very specifically, maybe even go after antibiotic resistance genes directly. A big deal for health.
Yeah, much more targeted than broad antibiotics. The notes also mention gene drives
Ew.
for pests.
Right, gene drives. Fascinating. But also requires real caution. It uses gene editing to push a specific trait through a whole population quickly, like making mosquitoes unable to spread malaria.
What the risk?
The risk is unintended consequences. Once released, it could spread uncontrollably, impact ecosystems in ways we don't foresee. So, massive need for careful risk assessment and, frankly, public discussion. It touches on dual use concerns, too.
Definitely sounds powerful and potentially risky. Any other agricultural applications?
Food safety, too, like using gene editing in chickens to remove the genes from major egg allergens. Think ovomucoid ovalbumin. That could be life-changing for people with allergies.
Wow. So from crop monitoring to animal health to food quality,
Mm.
a lot going on. What about energy? Biofuels came up.
Yes, biofuel production is another key area, reducing oil dependency, right? Synthetic
Right.
biology helps design microbes, bacteria, yeast to be better biofuel factories. By engineering synthetic expression systems to optimize the enzymes needed to make fuel molecules. Also exploring things like consolidated bioprocessing, CBP, trying to combine breaking down biomass like plant waste and turning it into fuel all in one step, in one organism, making it more efficient, cheaper.
So engineering microbes to be super efficient fuel producers from waste materials. Cool. Now, what about the environment? Bioremediation was mentioned.
Absolutely. Using biology to clean up pollution. Synthetic biology offers powerful tools here.
Such as?
Well, first, detection. Developing better biosensors. Our sources mention sensors engineered to detect specific pollutants like heavy metals, mercury, fluoride, and water, maybe giving off a light signal or changing color.
Living pollution detectors.
Kind of. And then the actual cleanup. Engineering microbes or plants phytoremediation to actively remove or neutralize pollutants.
Phytoremediation, using plants.
Yeah, different techniques. Phytoextraction pulls pollutants into the plant. Phytostabilization locks them in the soil. Rhizofiltration uses roots to filter water. Phytovolatilization releases them harmlessly into the air. Synthetic biology can boost these natural abilities.
Can you engineer organisms for really tough cleanup jobs?
That's the idea, using CRISPR to modify extremophiles organisms that live in harsh places to make them better at bioremediation in really contaminated or difficult environments. Or designing genetic circuits in microbes to boost their ability to break down specific nasty chemicals.
So better detection and better cleanup solutions. That's hopeful. We also saw a brief mention of medicine insulin production being a classic example.
Right. While our main focus today wasn't medicine, it's foundational. Producing recombinant insulin in bacteria back in the day was a landmark synthetic biology achievement, really, using engineered organisms to make life-saving drugs.
And the potential is broader now.
Oh, definitely. Things like new diagnostics, targeted drug delivery, maybe even artificial cells for therapy. It's a huge area, though not detailed in these specific sources.
It really shows the breadth. But with all this power to engineer life comes a responsibility, right? Safety, security, ethics. Our sources touched on this quite a bit.
Absolutely critical. Dual-use concerns are a major theme. The potential for these powerful technologies, even if developed for good, to be misused accidentally or on purpose. That poses risks to security, health, the economy.
So how do we manage that? The U.S. and China discussions mentioned governance.
Yeah, there's a clear need for strong oversight mechanisms. That means laws, regulations, but also softer guidelines, best practices within the research community itself. It needs to keep pace with the science.
And communication is key.
Crucial. Dialogue between researchers, industry, government, especially around funding potentially risky research and balancing transparency with security needs. How much do you share? How do you prevent misuse? Tough questions.
And beyond safety, there are ethical debates too.
Big time. What's the moral status of synthetic organisms? Does this change our understanding of life? How do we even talk about it? The metaphors we use matter. Plus, ethics of knowledge, especially dual-use research, and balancing the hype, the promise of benefits, with the need for caution, the precautionary principle.
It sounds like fostering a culture of responsibility is important, maybe starting early, like the IGM competition prioritizing safety.
Exactly. Initiatives like IGM are great models for training the next generation of synthetic biologists to think about safety and responsibility from the very beginning.
Okay, let's try to wrap this up for you, the listener. Synthetic biology. It's clearly a dynamic, powerful field. It's pushing biological boundaries, offering amazing potential solutions for agriculture, energy, the environment.
But, and it's a big but, that power demands really careful thought about safety, security, and the ethical side of things.
And here's where it gets really interesting for you to ponder. This ability to design and build life from the ground up, It opens incredible doors, but it also forces us to ask deep questions about our relationship with nature, our responsibilities.
Yeah, it's not just science, it's philosophy, it's societal impact, all rolled into one.
So as you explore new fields, keep an eye on areas like synthetic biology, where cutting-edge science, societal impact, and ethical challenges really collide. Staying informed is key to navigating what's coming.
Definitely.
And maybe a final provocative thought for you to mull over. If we can engineer biology to solve huge problems, are there limits to what we should create? And how do we make sure these tools are used responsibly for everyone's benefit?
That's the billion-dollar question, isn't it?
It certainly is. Thank you for joining us on this deep dive into the fascinating world of synthetic biology.